So yeah, my topic is genome editing for glycogen storage diseases. Here are my disclosures, mainly around a clinical trial that I started with the company AskBio. And in my outline, so I'll talk a little bit about an unmet need. Dr. El-Garbawi kind of mentioned that new therapies are needed for GSDs, and some of those might be around genome editing, as I'll explain. I'll talk about different approaches to genome editing, mainly for GSD1A. And here's my risky attempt at a joke. We need to settle a controversy like Arabic versus Roman numerals, a space or a dash. What are we going to do about this? What do we call GSD1A? Sorry, nerdy joke. No one got it. Thank you. And I'll mention Pompe disease. As you heard, but showing a little in detail, we've got this understanding of what causes a genetic disease. And there's this hierarchy from DNA to RNA to protein. And this underlies a genetic disease like a GSD. So what if you have a mistake or a pathogenic variant in your DNA? And so you don't make the RNA, you don't make the protein that are functional. And if you have two variants in a gene for a GSD, you'll have that condition. So a brief review of the genetics. And that leads into how to treat these conditions. So with gene therapy, this diagram shows how a modified virus, AAV vector, can bind to a cell, travel to the nucleus where the chromosomes are and the genes are, and then unwrap, and it'll be a functional DNA there. It makes these rings called episomes, just sitting there. They're not in the chromosomes, they're sitting in the nucleus, but they make RNA, they make protein to treat a disease like a GSD. In the liver, if the vector goes to muscle, it'll make the protein there to treat it there, also for the heart. And one key thing is this episomal DNA, like I said, is just sitting in the cell. So if the cell divides, you can imagine that'll be diluted out. The cell's not making more copies of the episomal AAV DNA. It's just sitting there based on the original treatment. And then a short course, a little bit more about the mechanisms or the structure of these vectors. Again, a modified virus like AAV. It has DNA, what we call a vector genome. Inside there's a transgene or therapeutic gene. This has pieces like a promoter's on-off switch. It determines where the gene is active, a cDNA that codes for a protein, and other details that help the transgene function. And the only AAV part of these little sequences at the end, the terminal repeats. But it's all packaged as this vector particle. And it acts just like a virus, so it's going to go into the tissues, like I showed you, like the liver. And then there's two approaches. So we could give a simple IV. This is used more commonly. It goes to the blood, to the liver, potentially to other tissues. Another entirely different approach to gene therapy modifies stem cells that can be injected, and they'll go to various places in the body, probably with lower efficiency. And then a problem of treating early in life. So here I'm just showing the liver. But we can give a dose of the vector. It'll go to the liver. For an infant, obviously, there's a much smaller liver, and we don't understand fully, is all that virus taken up by the infant's liver? Say you gave the same dose even that you gave an adult, will it be just concentrated here? If it were, when the liver grew to normal size, it would have the same amount of vector at the end. None of it was lost. It just stayed in those cells divided at the end. It should look just like as if you treated an adult, but that's not what we see in actual experiments. This dose of AAV, of gene therapy at the beginning, is actually lost over time when you treat early in life. And that has to do with the cell division, also maybe active elimination of the vector in the liver, even through normal processes of how a body reacts to a virus. So the issue is treating early in life like we need to for GSDs and having a long-term benefit. So that's one thing we attempt to address with genome editing. Genome editing makes a stable change in the DNA, while AAV gene therapy is generally lost. Genome editing attempts to correct the disease gene changing the DNA in a gene that will permanently correct that cell as long as the cell lives, and that cell can divide and the daughter cells will still be corrected. So it's like this picture again where there's a dose in the beginning for the infant and it's stable and it lasts to adult life. That'd be the goal of genome editing. Some of the details, you hear about CRISPR-Cas9. That's an RNA called the guide RNA plus a protein called Cas9. It makes a double-stranded break in the DNA in one particular spot. And there are two possibilities from there. And so our goal is gene correction. And in that case, there would be what's called homology-directed repair to precisely insert some DNA and have repaired DNA that's corrected. Another possibility that's a lot more frequent, actually, is what's called error-prone repair, introducing little mistakes at that break because it's error-prone. That's gene inactivation. Not what we want, not our goal here when we're thinking about correcting genes. It's more frequent. It has actually some potential therapeutic applications, like this can be used if there's a toxic gene you want to shut down, but not for a GSD. So we've already done a study in our dog model for GSD1A, space, Roman numeral. And what we found is kind of what we expected. Stably corrected hepatocytes where the gene was edited, shown by the little hand with the pencil here. So over time, actually over 16 months for these treated dogs, the cells that were edited stuck around very stably. The episomal AAV, as expected, is gradually lost. So we ended up with a fraction of the cells that were stably corrected, about 1%. But that gave us a starting point, and we're moving on from there, knowing that this can work in principle. And so let's step back a little bit and talk about the disease and what we need to treat. There are various estimates about how frequent GSD1A can be, up to 1 in 50,000 births. We understand the deficiency, the glucose 6-phosphatase enzyme, and it's, as we understand, responsible for breaking down glucose 6-phosphate into glucose, which is so very, very important. The liver is the main organ responsible for producing that glucose, so that's the main target. And the initial problem, of course, is managing hypoglycemia. So normally, the glucose 6-phosphate broken down into glucose goes out into the blood and is, the normal situation. But in GSD1A, that's not possible because of the absolute block here to making glucose. So the glucose 6-phosphate has nowhere to go. It accumulates in the liver, in the kidney, and it causes all those biochemical abnormalities that Dr. El-Garbawi detailed earlier. We've got animal models, starting with a mouse, and so these are mice that have 2 mutant copies of the gene, the G6PC gene. They have a severe phenotype. You can see here in the picture, this is a GSD1 mouse next to its littermate, and so they don't grow well, they have a large liver, they have the hypoglycemia. They don't survive past weaning without a gene therapy, without something to treat them. And then they have the involvement of the liver and kidney, and you can kind of see and imagine the liver's enlarged and full of glycogen here, same for the kidney. And we have to treat early in life to promote the survival. However, we have multiple endpoints to show benefits. So all these tests we could use to show if a therapy is working. And the canine model is similar. Again, this problem. So how do we understand the problem for gene therapy in GSD1A? And the AAV particles are going into the liver. There are these episome vector genomes that are sitting there next to the chromosomes, gradually lost when the liver grows and divides. And a couple of the research groups have shown that there's a greater than 90% loss of benefits from gene therapy in GSD1A treated early and then followed for the first year. And you can see in the staining of the liver that at 6 months, actually, there's plenty of brown staining across the cells in the liver, and it's lost by 12 months as the mouse continues to lose the episomal AAV. But our hypothesis, again, our theory is that genome editing will insert the therapeutic transgene to stably treat GSD1A. And we've been comparing gene therapy to genome editing. So here's our approach, our new experiment. And we've got two AAV vectors. It's just a way to deliver these tools. Could be a different vector, could be partly with RNA, but we know we need a vector to deliver the DNA. So here we're delivering the Cas9 with what we call a CRISPR vector, the G6PC or therapeutic gene that can insert into the chromosome as a donor vector. And also there's a guide RNA. And how this will work, again, it's the double-stranded break here in the G6PC gene. So, it might inactivate the gene, but it's already inactivated. So we can safely insert a therapeutic gene there. And then the donor, it pairs with this homologous sequence, and then through homology-directed repair, it's inserted here. And this is a functional gene that's at the beginning of the mouse's G6PC gene. So this could treat any mutation, could treat GSD1A regardless of the mutation. And here's more about our experiment. So side by side, we give the CRISPR and the donor here for genome editing, like I've been talking about, or the donor alone without the CRISPR. It won't insert; it'll stay as episomal DNA. So that's gene therapy. On the other hand, this episomal donor actually makes the enzyme, so it starts treating our mice immediately, and that's an important feature of this genome editing. There could be other approaches where you don't deliver a therapeutic gene and you're not treating the mouse until there's the genome editing occurs. So we've got an early treatment and then a long-term treatment when the genome editing happens. And then here we've got our little mice, infant mice. They're about 12 days of age. Their eyes are open. They have fur. They're not like a neonatal mouse, which I think is not the best model. These are a lot more like human infants when we treat them. And then they grow up to adults, and we look at the effects after 12 weeks. For this experiment, we've also added a drug for some of the groups, bezafibrate, which we know improves the response to AAV in GSD1. So we've got drug plus genome editing in comparison with gene therapy. Lots going on, but it helps to show how the genome editing is working. So how did it work? Well, here's first of all, our survival curve. I think you've seen these before, but as these curves go down, it means that the mice in that group are dropping off. And so when we did the Cas9 only, the CRISPR alone, we wouldn't expect that to treat GSD1, but it's an important control, and it didn't. So these mice didn't go past weaning. Then up here, if we give some combination of donor plus CRISPR, the genome editing effects, good survival, especially after the initial period, straight out here. But if we give the donor alone, and that's the gene therapy treatment, without our beneficial drug, you see how it continues to drop off. So here's some improvement with our genome editing. Got it. And just to summarize that precisely, we need either the CRISPR or the bezafibrate to get the best survival in this experiment. Looking back, well, a little bit more about this drug I've been mentioning. And in context of Dr. Elgarbawi's talk, what it does is it increases the use of fat for energy. That's how this bezafibrate works. It increases the so-called fatty acid oxidation. And that makes the cells healthier in the liver. We can see how they respond better to our genome editing, actually a previous version of genome editing with the drug added compared to without the drug. Here, all the brown is the enzyme, and it's much closer to the wild type than without the drug. We also know that this stimulates a particular cellular process called autophagy. We know that's important to treating GSD1. We also know it's important to helping the AAV vectors to work. So this drug plus genome editing is a kind of a combination therapy that can improve the treatment. And so we look at glucose, because glucose during fasting is really important. We fast the mice 8 hours after they're treated. At 2 weeks, all the treated groups were doing well. It takes a little bit longer to tease out, the actual differences. And at 11 weeks, then the group that is donor plus CRISPR plus bezafibrate, our genome editing plus the drug, did best, and that's shown here. It's improved compared to all the other treatment groups not quite as good as a normal wild-type mouse, but certainly doing better. And that explains also some of the improved survival. The liver glycogen was down. Actually, first, the enzyme activity was 8 to 10% of normal, and that's kind of the level that we need for a nice benefit. And again, our best treated group here in the red box starts to look better compared to, say, low-dose groups that we also treated, and untreated mice, well, mice that got just the CRISPR. Certainly the glycogen content is much, much lower. A little bit above normal, but still a good treatment. And then we can look at DNA and RNA for the therapeutic gene. This is really important to understanding what's going on. And the DNA is higher than other treatment groups here for our best treatment, and the RNA is higher as well. RNA higher than all the other groups, and just confirming that this combination is working well. Another feature is the Cas9. So we showed we don't need to use that. We don't need it around to treat the disease. It's actually just a tool for the genome editing. We don't want it around long-term because remember, it's cutting up DNA. Even in that specific spot, we don't want it to be, cutting DNA, and there's a chance it could cut DNA at other spots. There's a risk of what's called off-target effects, making DNA breaks where it shouldn't. But we're showing here that between day 3 and day 12, that CRISPR protein, the Cas9, is disappearing. It goes down 120-fold, and it's present in about 1 in 50 cells, or 1 in 5 cells here at week 12. But it's encouraging that the CRISPR is disappearing and our therapeutic gene is sticking around. We also, at the DNA level, could show that there is genome editing, that this just shows a PCR band that we could sequence that and show this is our gene inserted exactly as we predicted, precisely inserted, and that goes up when we add the drug. So we're seeing the integration, the insertion of the therapeutic gene going from 3 to 6% when we add the drug to genome editing. Also looking at the off-target unintended effects, is the CRISPR cutting elsewhere? And we looked at 12 predicted sites that are very similar to our target site, and we didn't see it cutting elsewhere. That would be if the open circles were higher than the red circles, and really they're not at these 12 different sites. So that's encouraging for safety. Now let's put this into perspective a little bit. My group is far from the first to work on genome editing, even for GSD1, and there have been other projects. So actually Dr. Janice Chu did a genome editing for the RA3C mutation with a similar strategy, and she had about 0.7%. In our dog, we had about 0.5 to 1% editing. Other studies done in mice with other genetic diseases typically have about a 1% editing efficiency. It can go higher in diseases that are toxic to the liver if you go long-term. And we can understand that in diseases like methylmalonic acidemia or tyrosinemia, other metabolic disorders. There's toxicity to the liver cells. Treating them with genome editing corrects those cells. Those cells have an advantage in growing over the long term, and so the benefits can go up over long term as those healthy cells that are corrected populate the liver. And we may be seeing that kind of thing in GSD1 because in our mouse we're at 3 to 6%, which is certainly above average for the field. GSD1 might be a little toxic to the liver, and we're correcting that, and it helps our therapy. Just to summarize then, up to 6% of the integrated transgene, 8% of the enzyme, 8% of normal. We know from historical studies that above 3% will be very therapeutic. Our vector dose, I hadn't mentioned, is about 5-fold lower than for similar studies, so it's working well. And then we've got a diagram to kind of talk us through, summarize. So the gene therapy arm, again, no CRISPR-Cas9, episomes in the liver cells gradually lost and less benefits in the disease over time. With the Cas9 for editing plus the bezafibrate, stable insertion of the therapeutic gene, and healthier mice in the long term, better outcomes, less hypoglycemia. About the point of how much enzyme. So a threshold of 3% prevents liver tumors from GSD1 in our mouse models. And a lot of this work is from Dr. Janice Chu's group, showing very precisely we need 3% of normal to prevent those liver tumors. And so we're at 8%, and that's looking encouraging. Now, a little bit back to the dog model, and we're doing more experiments. And with an improved approach from what we've learned in the mice. We know historically, these two papers shown here, actually from Florida group, from Duke, that to treat these puppies with GSD1, we'd need to use multiple AAV vectors over time. We'd have to switch the AAV each time because when you give an AAV, there are antibodies against that one, and they would prevent any benefit from using that same AAV again. But we have multiple types of AAVs, so we can treat again over time with different AAVs, and that's what's been necessary. And it had to happen in the first 2 to 4 months to actually stably treat these puppies who got the gene therapy first at birth. Genome editing then could avoid that, potentially, again, a stable correction. And here's our new study where we've treated 3 littermates with GSD1. And what's encouraging is that the glucose during fasting is improving over time. So we take away the food for the puppies and we check their blood glucose every 2 hours. When it drops below 50, then we stop and feed them. And this just shows how, early on after treating them, after 2 weeks, they were fasting 6 to 8 hours. At 3 months, maybe as predicted, they hit a low point here and couldn't fast 4 hours. And then without an additional treatment, it's coming back up again to 7 hours at 5 months. So this is what we predict if the genome editing is actually working. And this is just another way that we measure the glucose, showing it's in the normal range now at 5 months. The dose is higher. That's one thing that we've had to do, and this is probably more predictive of the kind of dose we'd need for treating human patients, if you will, in a clinical trial. So conclusions for the dog, the model, without going into more details, is going to predict better what we might need to do in a clinical trial. And we're able to do longer term studies, even though the dogs, age in dog years, they live multiple years, and obviously we'll learn a lot from a long-term study of these puppies. And then further things we need to do to move this ahead, we need to study how genome editing works in human cells. Of course, they have human DNA, so it'll be slightly different with regard to the genome editing. And there are ways to do that. Patient cell lines, mice that have been transplanted essentially with a human liver, and we hope to do all those experiments. Switching, the BEAM approach, and so these are just slides from the website of the company, but it's public information. And it's a different way of genome editing, and it's base editing to correct a particular pathogenic variant. Like here, the R83C variant, and what's needed is to switch an A in the DNA to a G. And so they have an editing tool called base editing. It's delivered, it's RNA delivered with lipid nanoparticles as an IV, potentially to patients, not yet. But here in the newborn mice, this is their survival curve. It looks quite good with a dose of the base editing to correct the pathogenic variant in the R83C mice. It's given to neonatal mice with a good long-term survival. And then just to summarize again the public information, the mice live a year, they have a smaller liver, they have improved G6Pase, enzyme activity in liver, they can maintain normal glucose levels, and so good improvement, good benefits here. And next step for the company, as we know, are to file an IND to do a clinical trial. So overall, I want to try to sum up now for GSD1A. And so what we knew about gene therapy is well established that it can have benefits, even treating practically at birth can last for a year with gradually diminishing benefits. I would say for genome editing with CRISPR-Cas9, the data are showing that the benefits can last longer and more stably, similarly for base editing. If we think about efficiency, the efficiency of gene therapy is quite good at the start. But over time, genome editing will have a higher efficiency, and that's what our comparison is showing us increased benefits with genome editing, and similarly for base editing. I mentioned how there are issues with the immune response to AAV, the patient making antibodies. We can't use that same AAV again with any expectation of benefits. So that's an issue for gene therapy, for genome editing, as we've done it to deliver a therapeutic gene. For base editing, you probably get around that. So it's a question mark if there's no immune response, and maybe you could even do repeat treatment for base editing. So, I'm acknowledging there are different approaches. And then how applicable is this? How available will this treatment be? So, as it is, so gene therapy, genome editing with a therapeutic gene could treat all patients, actually all patients who don't have an antibody against the AAV. Maybe the antibody could be depleted to allow all patients to be treated. Base editing will be delivered for one, or developed for one mutation at a time. And each one of those in the current situation is a new drug to go through FDA approval after clinical trials. So you could see here, you can choose your strategy. And actually the great thing is that these treatments are under development. It's very hopeful. So finally then about Pompe disease, just a word. We've been working towards genome editing for that. And here the deficiency is a lysosomal enzyme, GAA. There's accumulation of the glycogen in the lysosomes, mainly in the muscle cells here, that interferes with muscle function. There's weakness. In infantile Pompe disease, there's heart involvement. And we have enzyme replacement therapy, gene therapy being under development. Still, there will be this issue with really treating in infancy effectively. ERT has its limitations, and then a standard gene therapy treating in infancy won't last. So we've been working on a genome editing strategy, and this will look similar to the diagrams I've shown you for GSD1, but here in Pompe. So we'd have a double-stranded break, and then our donor vector could integrate with the therapeutic gene. And so how does the data look? And it looks quite encouraging. So even after 60 weeks, over a year, in a muscle like the quadriceps and other muscles, we have more than the normal amount of GAA, and it's safe. We know that having extra GAA doesn't hurt. And then even after 60 weeks, over a year, the normal amount of glycogen, similar to a normal mouse here, and that's even without genome editing. That's just the donor vector. So we're still studying that. We're sorting out what you need to treat Pompe disease successfully in infancy. I will note that this is at higher vector doses, like are needed for genome editing. And so that's a little disclaimer, but it looks very promising if we can actually deliver the gene to the muscle. I'm going to mention colleagues here. Dr. Kishnani, who you'll hear next, has been so supportive through all this. Dr. Y.T. Chen, who I think you heard earlier too, started a lot of this research, and people in the lab and the clinical team, funding from NIH and collaborators, and just that's all I have. So, thank you.
Any questions?
Good morning. In regards to the 1B population and gene editing, would you say that it's going to be a little bit harder because they need to treat each mutation separately? So if one patient has this type of mutation and then another one has a different one, could you speak a little bit about that?
Yeah, the challenge is, and you've been hearing these mutation-specific treatments, they're efficient, but then each one requires new clinical trials in the current situation. So for GSD1B, there's not such a common variant like the R83C for GSD1A. That is a statement, I think, that it'd be more effective to use a therapeutic gene approach that could treat regardless of mutation. The hurdles are in GSD 1B. Historically, we would have thought we had to treat the blood cells because they're involved. But now with drugs to relieve the effects in blood cells, it's actually very encouraging that we could focus on liver and kidney in GSD 1B, just like GSD 1A. I think the focus has been on 1A because we had tools earlier, there are more patients, but the development of new therapies for 1B is right on the tails of 1A, and there's a lot of potential there. But a treatment that is useful regardless of the pathogenic variant, probably more important for 1B. I didn't quite hear that.
Which would be the gene therapy as opposed to gene editing?
Well, genome editing includes inserting a therapeutic gene, like we've been doing. So it'd be a stable effect, and it could treat all patients. The problem is it's a little less efficient at this current time.
Hi. I was wondering if bezafibrate on its own could be like a possible therapy for GSD1A.
Yeah. Now in our mice, we treated for a week and shown that the liver glycogen and triglycerides go down. It's not enough to prolong survival, so we still need that gene to replace the enzyme. But it's a helpful additional treatment. Yeah.
Could that be helpful in humans to help reduce triglyceride levels at least, or prevent hypoglycemia?
Potentially, we need more information. There are a couple of case reports actually of prescribing a similar drug, fenofibrate, that's FDA approved in the U.S. The kind of things would be, potentially to lower triglycerides because that's what it's doing. It's burning fat. So, and it, has to be used carefully and watching for any side effects. But it's a potential treatment, co-treatment. Yeah.
Thank you.
Anything else, or should we get back on schedule here?
Thank you, Dwight.
Okay, you're welcome.